Yeah, so thanks for the opportunity to present some of my data today. So I'm quite excited, and I'm happy to receive any questions and comments and discussion points at the end of this talk. So most of my talk is going to be talking on an age-related tau pathology that occurs in astrocytes. So Lars has given a really nice introduction about the function and a bit about tau, quite a lot of information about tau pathology in neurons. And this is a very quite recently described age-related tau pathology that occurs in astrocytes, and it was only consensus recommendations were only published for this entity 5 years ago. And I'm going to talk about this prevalence of this age-related pathology in a longitudinal aging community-based cohort. So just as a very quick overview about age-related pathologies, it's actually well established now that these age-related pathologies are quite prevalent and a lot more common than previously assumed in the brain. And it's been documented in very large autopsy studies that there's a very complex constellation of age-related pathologies that are observed in individuals, particularly over the age of 65 years. And these predominantly occur with the deposition of the proteins tau, TDP, A beta, and alpha-synuclein. And of course, these are the four most commonly deposited proteins in neurodegenerative disorders. And as Lars touched on, of course, tau, in particular neuronal tau, is the most commonly deposited age-related and also neurodegenerative protein. And this is one of the famous studies by Brack and colleagues that Lars mentioned, and we can see that almost, or by the time individuals reach their third decade of life, almost all individuals will have some type of tau deposition in their neurons. And of course, the prevalence and the severity of this increases with advancing age. So all of these age-related pathologies have been reported in very large community-based studies and also studies in neurodegenerative cohorts. And it's generally assumed that these age-related pathologies really lower the threshold or lower the individual's threshold for developing cognitive decline and impairment. But of course, these age-related pathologies are also observed in the absence of a clinical phenotype. But the prevalence of these age-related pathologies in individuals with and without cognitive impairment really suggests that these proteins inevitably occur, or I should say the deposition of these proteins inevitably occur with normal brain aging, independent of neurodegenerative disease. So how we classify age-related pathologies is actually really similar to how we classify the neurodegenerative pathologies. So these are just some example images of the different neurodegenerative proteinopathies. And for the age-related pathologies, we are also looking for the presence of the intracellular or extracellular protein deposits in discrete and anatomical regions and very specific cell types. So this is just a sort of very overview summary of how we classify neurodegenerative diseases. And of course, some of the age-related pathologies are tucked into this classification scheme as well. So if we just look at the top coloured panels, they're coloured by the main protein deposited. So the tau proteinopathies, there are a number of different types, depending if it's a neuronal predominant, if the tau protein's in neurons and glia, or a glial predominant. Same with the alpha-synuclein and same with the TDP. But hidden along here are these age-related pathologies associated with the tau protein. And it's very interesting that we have neuronal predominant age-related pathologies, a mixture of both neurons and glia, and then this recently described age-related pathology in astrocytes. And same with alpha-synuclein, incidental Lewy body disease is commonly observed in large autopsy studies, and it's also described as an age-related pathology as well. And similar for TDP, this age-related TDP pathology was only recently described, I think, two years ago, and I'll touch on some of this pathology at the end of my talk. But my talk today is mainly going to be focused on this aging-related tau astrogliopathy, and that this is a pathological entity that was only described 5 years ago, and we've done a fair amount of work on this pathology at the moment. So this pathology is quite unusual because Lars has told us all about, the function of tau in neurons, and it's quite well established, but why tau is in fact in astrocytes and why it abnormally accumulates in astrocytes is very poorly understood, and we're doing a few studies to try and sort of work out why this might occur. But in large autopsy studies, it's been commonly reported and found that there's, these astrocytic tau pathologies throughout different brain regions. And this was commonly observed in individuals over the age of 60 years with and without cognitive impairment. So this entity was described as or named aging-related tau astrogliopathy, or ARTAG, in 2016. And this consensus paper describes how we classify and also standardize this age-related diagnosis. And it was based on the observation of these different types of astrocytic inclusions in different brain regions. And this age-related tau pathology in astrocytes is actually very common. So in other autopsy studies of patients that have come to autopsy, it's been reported to occur in over 60% of patients with dementia, and even over 70% of patients with a movement disorder. But I must say the prevalence rates, there's a slight selection bias, because a lot of studies, when they're looking at these age-related pathologies, they have very specific disease or neurodegenerative or control healthy groups. So the prevalence of some of these age-related pathologies in longitudinal aging studies and the general community is less well established. But the consensus paper described different types of this age-related tau astrocyte pathology, because it's found, in different brain regions like subependymal regions, subpial, around blood vessels, and in the white and the grey matter. And this all helps with the classification of this disorder. And we've also done a number of follow-up studies, but it's quite unusual because you can get this age-related pathology in many regions throughout the brain. And this is just some of the cortical and subcortical brain regions that we've mapped the distribution of this age-related pathology to. I haven't shown the brainstem diagrams here, but it's quite interesting because, of course, because this pathology can be found in so many widespread and anatomically diverse regions, you really need to discriminate these pathologies from the main neurodegenerative proteinopathies if you're looking. And we've also done follow-up studies where we can recognise the different types of age-related tau astrogliopathy from other proteinopathies. We're pretty good at recognising the types, and we've done studies looking at the different severity and anatomical regions where this pathology occurs. But just leading up to a study I have been working on, because we sort of wanted to get a better idea of how prevalent this pathology is in a community dwelling aging population. So we wanted to investigate the prevalence of cortical age-related tau astrogliopathy, and we had access to a large European community dwelling population. And we also explored the regional overlap of the different types of this pathology. So my collaborator and I were really lucky because we had access to the Vienna longitudinal community-based aging cohort. And this is where all participants born between May 1925 and May 1926 on whatever district on the left side of the Danube River were enrolled into this study. And the other year when I was in that lab doing this study, we were lucky because at the time, well, it was fortunate that we had access to 310 cases that were available for the pathological assessment. So it's an aging study, so the mean age was 83 years, and we had a slightly higher rate of females at the time. And we looked in the frontal, parietal, temporal cortices for sections immunostain with phosphorylated tau. And of course, we found multiple types of the different aging-related tau astrogliopathy in our cortical brain regions. Some of the images are just shown here. So the very characteristic tau inclusions in these astrocytes, which is, I must say, quite nice to see. So we're actually quite surprised because we found that age-related tau astrogliopathy was actually very common in this longitudinal aging study. So it's almost 40% of all cases had some type of this age-related tau pathology in astrocytes. So if we separate the different brain regions, these are the cases with this pathology. We can see straight away that the grey matter type of this pathology was most common compared to other types, particularly the perivascular type. But we did another fair amount of pattern analysis, and we found that, yes, while 57 cases had this type of pathology in one brain region, it was actually quite complex because a huge number of cases, well, 36 cases had this type of pathology in multiple or two brain regions, and 25 cases had this type of pathology in all the three cortical brain regions examined. And it actually paints a quite interesting picture when we're looking at these age-related pathologies in the human cortex. So this is all the patients with any type of age-related tau astrogliopathy in the cortex. We just sort of map the distribution. So up here, each row is the different cortical regions. So for example, these patients only had one type of this age-related pathology, but it actually gets quite complicated and quite complex because we had a number of individuals that had more than 3 types across multiple brain regions. And we had some patients or some individuals, with 7, 8 or 9 different types of these combinations of tau pathology. So I just finished these stats yesterday because we're now doing a follow-up study on 101 patients out of these 300 patients where we now have access to multiple brain regions. And this is including the medial temporal lobe. So that includes the amygdala and the hippocampus. And this brain region, I must say, is actually really important when we're looking at these age-related pathologies because it's for some reason, it's a known predilection site for these pathologies. And of course, we also had access to the cortex, subcortical structures, and also multiple brainstem structures. So this is just a summary of the different types of pathology we've seen in this longitudinal aging study of 101 patients. And this is the percentage or the prevalence of these pathologies in any brain region. But we can actually see that quite interestingly, and also I was a bit quite surprised, is that aging-related tau astrogliopathy, we're now reporting in a prevalence rate of up to 80% of individuals. And this rate is effectively double what we've found in the cortex because we know that the medial temporal lobe, which is included in this study, is a known hotspot. And then we've broken down the different types of age-related tau astrogliopathy pathologies. This is the bright neurofibrillary tangle stage. And if we add up all of these columns, it is effectively 101 patients will actually have some degree of tau deposition in their neurons. We found around 60% of individuals will have A beta plaques, 40% will have cerebral amyloid angiopathy. Around a third will have Lewy body pathology, and this is mainly found or localized in the brainstem. And we're also quite interested because TDP, or limbic age-related TDP encephalopathy, this is the recently described TDP age-related pathology. And it was only described 2 years ago, and we've already reported this now for the first time, or I'm writing in the paper, but it's actually found in 20% of this longitudinal aging study. So when I was doing some of the association statistics, we've actually found it's quite complicated because if you have one type of age-related pathology, you've got an increased chance of also having another age-related pathology. So this TDP, age-related pathology, is also associated with aging-related tau astrogliopathy in some types in particular. And it was quite interesting because then also the number of brain regions containing aging-related tau pathologies was actually higher in cases with this TDP pathology. We're also actually very surprised because we've also found quite unexpectedly that oligodendral tau pathology was actually found in around a third of all patients. And oligodendroglia are another main class of glial cell, and it's not as commonly associated with neurodegenerative proteins. And we're actually quite surprised that in this age-related study, we've actually found oligodendral tau pathology in around 30% of all cases. And of course, this pathology also related to some different types of aging-related tau and TDP pathologies. So there was a lot of associations between these different types of age-related pathologies, and these also remained significant even after we controlled for age, sex, and bright stage. So I think there's quite a few complex interactions happening here, which is quite interesting. So just as a quick summary, our study also validates others where we know that aging-related pathologies are actually more common than previously assumed. And this really suggests that this age-related pathology inevitably occurs with aging independently of neurodegenerative diseases. But I think the identification of these pathologies has really been facilitated by the combination of both histological, immunohistochemical and biochemical methods, because we can now detect these pathologies and see them, where a lot of the earlier studies were mostly relying on histological methods, and therefore we didn't actually see these very fine and discriminative pathologies. But it's interesting because neuronal pathologies have actually been recognised for quite a long time, but these age-related glial pathologies are now recognised, and we've seen this in astrocytes and both oligodendroglia. But it's a little bit complex because when we're looking at postmortem human brain tissue, we really need to carefully discriminate, what is an age-related pathology versus what's a neurodegenerative pathology. And this will then, add to the complexity of how we classify and diagnose neurodegenerative diseases, particularly when it comes to pathological thresholding and anatomical distribution of pathologies. And of course, I think these age-related pathologies, will have implications for concepts around, disease pathogenesis and how these age-related pathologies might interact with each other. And I think they're also going to paint quite an interesting picture when we're thinking about the clinical phenotype. There have been a number of studies that have suggested that in autopsy-confirmed patients, that individuals with a higher burden of age-related pathologies actually have a more complex clinical spectrum. But I think there's a lot of these studies are still in the infancy, and it needs a fair amount of work to build on this further. I think a lot of work with the preclinical models is going to be quite interesting, because currently most of these are still looking at one main protein, and at the moment, depending on the study, they're not really capturing some of the age-related pathologies that we might expect to see in the human. And of course, these age-related pathologies are going to have implications for biomarkers and stratifying patients for therapeutic trials when we get there. But I'd like to acknowledge Lars and my colleagues at the Dementia Research Centre, and my colleague, Professor Gabor Kovac, who I collaborate very closely, and of course collaborators with University of Sydney and the Medical University of Vienna, and of course the funding agencies. So I'm very happy to take questions and comments.
Great, thank you, Shelley, for that presentation. If I can get you just to stop sharing, and we can see your face really clearly. Thank you. So if anyone has any questions, please go ahead and pop them in the Q&A box. But I will get started for a couple of things that I was interested in, because I'd never heard of these age-related tau pathologies, because this is not my area at all. So I'm sorry if you did say this and I missed it, but are there then big individual differences in people's profiles? So, you know, we often think about some people, the way people age is very different, right? And so is that something that you have detected or something you expect to see, that for some people there will be more of this or more in some brain regions than others?
Yeah, it's very interesting because when we look at, you know, some, you know, neurologically normal individuals, it's so diverse because some individuals will have, you know, so much age-related pathology and some will have like minimal or nothing. So it's why some people have more, some people have less, but also it's very interesting what some people can deal with this potentially better than others. So it might, I think there's many different concepts to tease out here, but a lot of these age-related pathologies and what this means for the individual and also clinically, it's still in its infancy. Generally, the consensus is that these age-related pathologies and any mixed pathologies will lower the individual's threshold for developing further neurodegenerative proteinopathies or a cognitive impairment. And then we need to think about how we're diagnosing these, because, you know, like mild age-related pathology in, you know, X part of the brain it might be unlikely to have a clinical phenotype attached to that. But if you have a very, you know, severe age-related pathologies in multiple brain regions, I think that needs to be carefully considered.
Yeah, so we see this, you know, a similar thing in the brains of people who have got these neurodegenerative diseases, right? So some people can have, you know, a lot of, you know, plaques and tangles, and then it doesn't always match the clinical profile. So some people can look like there's a lot of neurodegeneration there and yet perform actually really well. And then you can have other people who, you know, who have the opposite effect. And I guess when we think about it in terms of dementia, one of the arguments that is put forward is that the reason for this difference between pathology and the clinical presentation relates to this concept of cognitive reserve, right? So the idea that for, you know, the cognitive reserve is sort of this, you know, it's about the connections that your brain actually has, but more importantly, the ability for your brain to sort of have alternative pathways and connections, to be able to problem solve challenges. So do you think that's maybe what's going on here too, that maybe cognitive reserve has got something to do with this difference in the pathology, Sam?
Yeah, it's going to be interesting. I don't know if I have an answer to that at the moment. I just feel like being a little bit cautious before I... but it's very interesting. I think there are a number of dimensions to this as well. And you know, a lot of these, it's quite interesting because a lot of these age-related pathologies, you know, occur around perivascular regions, like around the blood vessels, subependymal regions, and also the subpial regions. And then, you know, it's been also hypothesized, you know, that as we age, and also in neurodegenerative diseases, I think it's been shown that individuals do get blood-brain barrier abnormalities. So potentially that could be a point of entry, and this could be some sort of response to that. I think that's only a very small sort of snapshot of the overall picture. But I do think the cognitive reserve aspect is very interesting, because we do see even, you know, in the oldest old, like there have been studies in individuals in their 10th and 11th decade, so 90 and 100 plus, you know, where their brains are just amazing and there's sort of minimal pathology. And in those cohorts, I think the Alzheimer's disease neuropathological change sort of doesn't correlate as well with the cognitive symptoms that they may or may not show. So I think it's an interesting space to watch this further.
Yeah, no, definitely. And just one more question before we go to break. And I think you've said this, but I guess, because I don't, again, please, you know, forgive my narrative thing here, but these age-related pathologies of tau, if the tau protein degeneration, I think Lars was saying basically it impacts on learning potentially. That seems to be one way that it works. So is that the same thing here? So because these are also tau pathologies, is this what's being impacted? Is there sort of potential learning ability?
It could be. What we're particularly interested in, because the age-related pathology I spoke about today in astrocyte is quite interesting, because Lars spoke about a lot about the function of tau in neurons, and particularly the axon, and also in the dendrite, and there's also some nuclear functions of tau that have also been described as well. But a lot of my work is sort of also trying to understand why we might get tau in these non-neuronal cells, because there are a number of studies that have shown that the mRNA expression of tau in your glial cell, your astrocytes, and your oligodendroglia is actually incredibly low. So why is tau abnormally deposited in these cells with aging, and also in some frontotemporal tauopathies as well? So if it's either expressed at such low amounts that we're just not very good at detecting it at the moment, or if it's taken up and incorporated into the cell, we're not too sure. So I can't remember if that answered your question. I forgot your question.
No, but that helped to remind me actually when we're talking about tau depositing in different areas. So that was very helpful. All right, so I don't think we have any other questions at this point. So thank you very much. That was a really helpful presentation. And to both you and Lars, we've been teaching us a lot about tau this morning. So